Thank you. Thanks, Ellie. Okay, I'm going to talk slightly different, so I hope I'm keeping it simple enough for the parents so that it is a bit more technical in terms, but I try to make it sound okay. So what I'm going to do is talk a little bit about what we know about from the genetic side of things and what possibilities there might be to actually try to rescue the Prader-Willi phenotype or improve it at least. Now, we're working mostly with animals, with mice, where we can do a few things which we can't do in humans. We can control the experiments much better. We can make genetic variations in the mice, and we also can test potential drugs in there, which we can't always that easily do in humans. And one additional extreme valuable point with animals is, in comparison to my clinical counterparts, the mice don't lie to me on how much they have eaten. So just going back to this Prader-Willi locus here, and the mouse and humans are actually genetically very, very identical. So on a primary sequence identity, we have about 93% of the mouse gene are the same as the human's gene. So that's very homologous. But there's still a few differences, for example, here in the Prader-Willi locus in the humans versus the mouse thing. And that's obviously, if we use a mouse study, we have to consider that. There's some of them are these snort genes, which don't seem to be that important for this particular reason, which I go in a minute. But there are a few other genes up here that are in the humans but not present in the mouse. Just listing up some of the functions. So there's three major genes, this MK and 3 thing. It seems to be important for the hypothalamic pituitary gonadal axis, and that's one actually causing some of the central precocious puberty, which you heard earlier this morning, which could have an effect there. Then there's a C15, or very ORF2 gene, which is only produced in the testes and is actually produced not in an imprinting factor, so it shouldn't matter directly to the research we're doing. And then there are two other non-coding RNAs, this non-RNA1 and RNA2. This are also expressed in some of them. The PN1 is biallelic in the testes and kidney, but it's monoallelic in the brain. So it means it's only from the parental lineage active in the brain, but active in the others, in all cells in the others. The same with the PNR2, which is produced in the testes and is biallelic, so it shouldn't have any of them an effect. So the mouse model is actually quite a good way to check out what's happening in these Prader-Willi locus. So there are too many genes to talk about them all, so I'm going to focus on some which we have been working on, which are now been identified as this snort cluster, which a few cases of humans Prader-Willi symptom people have been identified. They only have some microdeletion in this area, which misses basically snort 116 gene, or another snort gene, and this IPW gene. And they seem to have very similar and almost identical classical Prader-Willi syndrome things. So the idea was this might be the really critical genes. Now, we can probably exclude the 119A because there's another copy of that outside here, so one wouldn't necessarily have a reason to believe that's important. I will mostly focus on the SNORD 116, but I also want to talk quickly about this IPW gene, which encodes a so-called, we call it a non-coding gene, so it's just producing a ribosomal part of the gene, but it doesn't actually produce a protein, which most other genes do. And what's been identified, and that gets a bit complicated, so don't look too careful, what I want to just point out is this type of imprinting on SNORD cluster genes was always thought to be just a typical Prader-Willi thing, but there's actually a second cluster of SNORD genes on a different chromosome, which is also imprinted, and that's on the chromosome 14. It's called this DLK1 domain. And some recent research has actually identified that this coding sequence, or the non-coding sequence from the Prader-Willi syndrome, can interact with a protein here called a methyltransferase, which might interact with the imprintings on this other chromosomal location and actually activate the silence gene on this one. And that's actually quite interesting. That's the kind of principle they have kind of worked out. that under normal function, this Prader-Willi gene interacts with that molecule and then kind of sets up the right expression for these two imprinted centers. This chromosome 14 mutation normally occurs as a UDP, so if you have two copies, in this case from the mother, then this function is disturbed and you lose the expression of one of these important genes. In the Prader-Willi situation, where you don't have this molecule that can interact with that, then you're losing some of this methylation on these proteins, which doesn't have any influence on the expression of the normal gene, but it increases the expression of some of the SNORD genes. Now, the people with these conditions have some features that are kind of overlapping with or similar with Prader-Willi. They have short stature, they have a low birth weight, hypotonia, motor delay, feeding problems in early life, early puberty, significantly reduced final height. And they also have some facial features, including the broad forehead, short nose, and a wide nasal tip. And the majority of the people have small hands and feet. They also develop some kind of obesity, or they are on the quite upper end of the BMI scale. For 25 to 30, which is already in the overweight, and there are some examples they are out in the 40s, which is clearly obese. So the Prader-Willi syndrome locus might actually play a role for other incidences as well. So there is probably a bit of wider things we have to think of and when we look at this problem more. Now, coming back to the SNORD 116, again, that molecule itself is quite complicated, and it's produced. There's a huge reading frame, or another reading transcription going on of a long non-coding RNA, and there are lots of copies of the SNORD116 gene, and they're located what's called an intron, and when this RNA is processed to make the normal gene, where this bits in the middle will be cut out, and this normally gets discarded. Then there's some special process where this is removed to just where the location of the SNORD is, and then it produces these very short molecules. Now, the function of the SNORD molecules, there are a huge class of it. What they normally do, they form this structure, and they have some kind of homology sequence here, which they interact with other RNA molecules, and they modify them. And there are 106 RNA molecules known, they're being targeted by that. But you can already see there's number 116, and there is one more. So there's some of them, SNORD RNA, don't have a partner, and we don't really know what they're doing. And that's the same with the SNORD 116. There's no real function known for it. Whereas the SNORD 115, which sits next to it, targets this 5-HT receptor, which is a serotonin receptor, which actually has been implicated in feeding, but it doesn't seem when specific deletions happen in the locus with the 115 gene that anything happens to or Prader-Willi-like syndrome. Anyway, it's getting more complicated with that, because as I said, there's one SNORD molecule per intron, but actually there are 5 introns. They actually have 2 SNORD molecules in there, and when that happens, then you get a different processing for these molecules. And one is that they actually then have SNORD molecules on either end, and there's a big piece of RNA in between. And the hypothesis is that this is kind of a sink for a molecule called FOXO2, which is a protein that it's needed to modify RNA when they get spliced. So sometimes you can have A, B, C, and it splices A and B and C, or it splices A and C together and leaves the other one out. So you're choosing between different parts where you're gluing together. Now, in the Prader-Willi, the idea is that there's none of this sink there, so all these molecules floating around, and they go all crazy, and then you get all the alternative splicing wrong. We have looked actually in our mouse model where we delete the SNORD for differences in alternative splicing, and we can't actually find anything really significant in there. So this is being done mostly in cell culture, so it could be a difference there. Now, SNORD 116 is actually massively expressed in the brain. So this shows an autoradiogram, and this is the area with the most darkest areas, the hypothalamus, which you heard a lot from it, and this is the strongest expression there. Now, in the mouse, SNOD106 is exclusively expressed in the brain. In humans, you find SNOD1 in quite a variety of tissues. So there's one difference between the mouse model and the humans, which we have to consider, that there's SNOD106 function in other tissues too. Now, one thing that really drives food intake in the brain is a neuronal population. You heard before, this is kind of MPY neurons or HRP neurons, and this is a mouse model where we just put some color into these MPY neurons, and this is when the expression level when they're normal fed, and this is when they overnight fasted. So you can see how massively this is upregulated and how much is driving. If you kill these neurons, only this particular neurons, there's about 5,000 of them in the archaea, you die. You don't eat anymore and you're dead. So we can actually do the opposite. We can actually artificially increase MPY in these particular neurons here in mice, and that's happened after 3 weeks. These mice are really obese, because if you give them free access to food, they just eat. That's the most strongest feeding drive you can get. Now, importantly, if we look at mice that are missing the SNOD106, just as we see that these MPY levels, which are here shown, going massively up. Okay, so they actually have an enormous drive to eat. So the lack of the SNOD gene actually drives the feeding phenotype. And vice versa, if we look at the SNOD expression itself, when we fast animals, the levels go down. Again, indication that energy status is regulating the SNOD genes, which is a real consequence of how much energy you have and how much is driving. And that's the kind of thing. If you look at the mice are viable and think that they have a lower birth weight and coming up slower, but then you can see if you normalize it to the same age here, they're increasing their body weight much quicker and eating much more in a period of time, same as, in a sense, Prader-Willi children when they're trying to catch up phase. So that's a very classical thing. What's not happening in this mouse model, and actually in none of the mouse models that are generated by some of these genetic variations, is the mice don't really get obese. So that's one of the real differences with this, and that's why probably, and that's kind of the interaction with this Prader-Willi locus, with some other locuses might be really important that we don't see in this case because it's not there. Now, there have obviously been now some ideas of how we can change maybe the function back to activity, and one of the common things in all the different variations of deletions or the UDP or just the smaller mutations, you still have the maternal chromosome in there which has the genes on there, it's just not switched on. So one way to do is trying to find ways to switch that inactive gene, allele on, or in the cases where we have some smaller deletions like the SNOD RNA, can we bring that back with another way? Okay. Quickly, there have been some studies done where they've tried to put in 1 copy or 2 copies of SNOD into other genes and tried to see if they can rescue it. There's not really that strong effect because there are here 2 copies, and you can see the expression is not that what it should be, particularly in the acute hypothalamic nucleus it's missing, so actually you don't see much of an effect in this particular mouse. But there's been other studies where they're actually trying putting another what's called promoter in front of the genes itself, which is kind of a switch. So every gene has a switch on or off. And you put a new switch in here and turn the genes on. And in this case, you can get quite a good increase in expression of the SNOD gene again. And you also kind of rescue that early onset delay in development, and it gets normally back in this mice. So again, the problem with this model is that they're using all different kind of models where it's missing, so it's hard to compare them. But this one seems actually rescues this retarded growth phase, which is very important. Now, just quickly, a few little other ones. We come back to this molecule, this G9R, which I mentioned before in the other imprinting center. So DNA comes in 2 more forms. In one way it's active, it's more in a kind of relaxed form where it has some of these so-called histone molecules being bound to it, but it's not compressed together, so that's when it's active. There's this molecule which puts some modification on these histones, then compacts them together, and then the DNA can't be accessed and can't be, the information stored can't be translated into RNA. And that's what kind of is one other level of imprinting or epigenetic regulation. So in this case, people have tried to modify this with a ligand to just block the activity of that and loosen that up. And they can see that they get some of the expression of the SNORD back. So there's one possibility here. And they also show that they're changing some of the this methyltransferase. I don't need to go into that. Yeah. They're using also a very severe model of mice, and they can improve the survival and increasing body weight in the early phases, but this is a really severe model, and the mice died relatively early. So, but there's some promising things happening. This molecule I kind of skip because the next speaker will talk probably more about things like that. There's also attempts for the Angelman syndrome to reactivate the things there. Here it's a different approach. Angelman syndrome is characterized by missing of one gene on the UPC3, and there's a long molecule synthesized over here, and that actually blocks the activity of that gene here. And so what you do is you use something called an antisense oligonucleotide, which binds to that specifically, and then that degrades the strain here and reactivates the gene here. And you can see that down here, so they can get some nice activation of the gene. So there's another way of trying to manipulate that system. Now, just quick, finally, our approach is actually we're using a viral approach. So we get a virus where we put this S-NORD116 in, and then we're injecting that into the brain, into the arcuate nucleus, and we can see here that it's dark staining, that's silver staining, where we overproduced or reintroduced the SNORD116 gene, and these mice lose weight after that. So SNORD1 higher levels reduce appetite and weight, so it does work. The difficulty with this approach, for at least in the mice, is because we can only get, if I just show back here, this is 0.04 millimeters. So in order to get the virus into the right position, it's like a 1 millimeter area you have to go for, and that's very close. So you have to be very precise. We have to use animals that are about 6 weeks old, where we can start doing that. In human terms, that's already a teenager. So because they are already sexually matured in that range. So we still get a nice response, but if we can actually transfer it much earlier, we would actually get a much better response. Okay, so I'll leave it here. Just thanking the people who have done the work, so mostly Chi is a neurosurgeon who helps us with all these precise injections and with the help of others, and some of other members prior, they're helping us with that, and also Serg Sokolowski from Florida, who are helping us with the viruses. Thank you.
Thank you very much, Herbert. That was fantastic, the way you covered all those areas, which does give us hope that something will come out of all that. So keep it up. Keep up your research team. Okay, now the next speaker is representing the Australian foundation, the Prader-Willi Syndrome Research Foundation of Australia. So I'll ask Marnie to come forward, Marnie Hewitt, and she, if I could find the correct, if I could find it and read it.
It's fine.
She's got a BSc and a PhD at the University of Sydney, so that should be enough, shouldn't it? So I'll leave it to you now, Marnie.
Thanks very much.
Thank you.
Okay, so thank you very much to the organisers for inviting me to speak, and I'd also like to acknowledge the traditional owners of the land that we're meeting on for this conference today, who are the Turrbal people. So we're very fortunate to be here on their land. So I think it's a really nice follow-on from Herbert's talk, because what I'm going to talk to you about is epigenetics, and if we can target one of those epigenetic modifiers that is keeping that maternal allele switched off to be able to reactivate. And if this might actually be helpful, not only for Prader-Willi, but also Schaff-Yang, a related disorder I'll mention. So as we've seen this particular picture many times before, all I want to remind you is that all Prader-Willi patients have one normal copy, the maternal allele, and so they have the copies, just like Herbert was just saying. They have these copies of the genes there. They're just not being switched on. So this is the picture of the mouse, because mostly my lab works in the mouse. And so here in the blue on the bottom is the paternal allele, which would normally be being expressed, and in grey is the maternal allele, where they're making UBE3A, but not the rest of the genes. So the imprinted expression is that you're only expressing from one of the parental alleles determined by the parent of origin. But because of this normal allele, the potential is for us to somehow switch on this normal maternal allele. But to be able to switch something on, we need to know how it's switched off in the first place. So the way that it's switched off is by mechanisms that are called epigenetic mechanisms, and this prefix epi just means that there's some layer of information on top of that genetic information. In this case, it's chemical tags that can be added to the DNA. These are methyl groups, but also to the proteins that are packaging the DNA, the histone proteins. And we know that these chemical tags are added on during oogenesis onto the maternal allele here, and they are including DNA methylation and H3K9 methylation for the histones. So if we can somehow target these epigenetic marks and remove them again, then this would be one way that you could actually reactivate the maternal allele. Alternatively, you can target the epigenetic modifiers, the proteins that are themselves sitting there on the maternal allele and switching off that maternal allele. So that's where I come in. So I've only really been working on Prader-Willi maybe for 2 years, but I have been working on this protein since we've known about it, which is called SMUCHD1. So I discovered this protein, SMCHD1, which I don't want to even spell out the whole of its name for you because it's a bit of a mouthful, but I'll call it SMOOCH for short. So I found it more than a decade ago, and we published it first a decade ago, and at the time we knew almost nothing about it. But what we now know through lots of studies is that it is one of these proteins that's important for switching off the maternal allele at the Prader-Willi cluster. But unlike other proteins, it's not important for the whole of the cluster, so it doesn't really have anything to do with the SNO RNAs that you've just been hearing about. It really is only important at this one end of the cluster. So here I'm showing you where it binds through this region. It actually binds to the chromatin. And the SMUCHD1 sensitive genes are MAGEL2, MECORIN3, NDN, and this mouse gene, PEG12. But this second half of the cluster is the regions where it's important. SMUCHD1 binds it in mouse. It also binds there in humans as well. Interestingly, I think there's a bit of a difference to one of the studies that Herbert was mentioning, where they were targeting H3K9 methylation here. The reason it's different is because when we have no SMUCHD1, we know in these animals that they have loss of imprinting. They have this maternal allele being switched on for MAGEL2, NDN, MECORIN, but SNRPen and the SNO RNAs are left alone, and that means that you don't inhibit the production of UBE3A. And this is really important because that means you're not going to cause Angelman syndrome by rescuing Prader-Willi. Obviously not something you want to do. So this is the protein SMUCHD1 that we work on. It's a very large chromosomal protein. It's a dimer, so you can probably see this nice symmetry that it has. And over the years, what we've been able to find is that not only is it important at imprinted clusters, the Prader-Willi cluster, but also on the second X chromosome in women, the inactive X chromosome. We have 2 X's, we only use 1, and so 1 is epigenetically silenced by SMUCHD1. It's really important in embryonic patterning, and this is involved in Hox gene regulation and also in other repeated clusters of genes which are found in humans and contribute to FSHD. So the questions that are really important for targeting SMUCHD1 to treat Prader-Willi, potentially doing that, are: is it of any use to target and actually reactivate MAGEL2, MECORIN3 and NDN? We've heard a lot about the SNO RNAs, but what do these genes actually contribute? Is it worth the bother? Will it work to actually inhibit SMUCHD1 in neonates or later in life, or is it only going to work if you don't have SMUCHD1 right from conception onwards? What's the risk of targeting SMUCHD1? Because if it's important in these developmental processes I just mentioned, embryonic patterning and also this silencing of the second X chromosome, is that going to be dangerous to do? And then how on earth do you target this particular epigenetic repressor anyway? What are we going to do to be able to target it therapeutically? So I'm going to touch a little bit on each of these and give you an update about what we're doing. So we heard a little bit about this before. So for MAGEL2, we heard about precocious puberty. So people with mutations just in MECORIN3 end up with precocious puberty, which, as we heard this morning, is not particularly common, but it is one of the phenotypes in Prader-Willi. If you have a mutation just in MAGEL2, what was discovered a few years ago by Christian Schaff and his team was that this produces Schaff-Yang syndrome, which used to be called Prader-Willi-like because of the highly overlapping symptoms in these patients, and some of them are here. So hypotonia, developmental delay, intellectual disability, autism, hypothalamic dysfunction, there's decreased oxytocin, so many, many overlapping symptoms. And although there aren't patients that have been found with just NDN or nectin mutations, in the mouse model they have neurodevelopment defects and also some hormone-producing defects. So there's quite good argument to suggest that these 3 genes alone, reactivating these 3 genes, could have some benefit, especially if it were potentially in the end combined with other therapeutics. So what's the effect then? I've told you we have embryos that have never had SMooCD1, but what about if you then delete SMooCD1 only later on once you've already developed? What's the effect of acute deletion? Because this is really what a drug would do. So in this experiment, we're actually setting it up for a different purpose in the first place. We wanted to know what SMooCD1 actually does at the chromatin, and I'll tell you about that in a minute. But in these embryos, we took the cortices, and from the cortex material from the embryo, we derived neural stem cells, and these are useful because you can perpetuate them in culture for a reasonable length of time to be able to do some nice experiments. And then we used the genetic models that we have to be able to delete SMooCD1, and we had paired matched samples where they had normal levels of SMooCD1, and that's shown here in the Western blot. And then at 1 week post-deletion, so they'd been deleted for a whole week, no SMooCD1 for a whole week, we then looked at gene expression, and we did a lot of other measures as well, but I'm just going to tell you about the gene expression by RNA-seq. So the first and really good bit of news was when you delete SMooCD1 in this manner, once you've already developed the embryo, Hox gene silencing, this what's really important for patterning, completely fine. The inactive X chromosome, completely fine. So that's good news because it tells you that it's not that all genes are going to be sensitive to SMooCD1 deletion, not all of its targets. But what we did see is if you look at NDN, which is expressed in these cells, here you'll see the expression from the maternal allele that is imprinted, and it's normalized to the paternal allele, the one that's on. And we can tell the difference between the two because of some genetic differences between them. And what you'll see is when we delete SMooCD1, the maternal allele is upregulated a bit, and this was kind of tantalizing. It wasn't formal proof, it wasn't quite enough evidence, but it suggested that this was going to be loss of imprinting, just like we wanted to happen, despite the fact we didn't affect the rest of the genome. We also didn't affect SNRPN, and we didn't affect UBE3A, which was promising. But interestingly, you'll note here, the maternal allele is expressed at a lower level than the paternal allele, but it's not nothing. So it's not particularly imprinted in the cortical neural stem cells. It's not completely imprinted. So we extended this experiment to instead make the neural stem cells from the hypothalamus, so hypothalamic neural stem cells in a new technique that hasn't actually been done before. And now you can see that we have to truncate the axis because now we have got proper real imprinted expression. And I think that's quite interesting for anybody interested in imprinting and how it's regulated like I am. Now when we delete SMooCD1, we get a much more striking increase. Again, it's not up to the same level of expression as the paternal allele, but we are switching it back on again. And this mouse-specific gene, PEG12, has quite a striking loss of imprinting as well. So then this is again good news. But what we don't know still is, so we still don't affect SNRPN or UBE3A, which is great, but we don't have expression of one of the critical genes, MAGEL2, the one that is mutated specifically in Schaff-Yang syndrome, the one Prader-Willi-like, one that we really care about. But it seems to be only expressed in hypothalamic neurons, not the neural stem cells, the cells that really aren't so great for culturing. So now what we're doing is taking the hypothalamic neural stem cells and differentiating them into neurons to see if we can get MAGEL2 to be expressed at an appreciable level, to see if we do get loss of imprinting. We're also trying to do that from embryonic stem cells and an in vivo mouse model where you can delete only once the pups are born, so in a neonatal setting, which might be actually most relevant. So we're keen to see whether this is going to still result in loss of imprinting and upregulation of this cluster. So how would we inhibit SMooCD1? We have some promising evidence, we still need to get more. Well, what would we do to actually therapeutically manipulate SMooCD1 function? So a couple of years ago, we were able to study this end of the protein, the N-terminus of the protein, and produce it as a recombinant protein, so produce it in vitro. And we know that it has ATPase activity. That means it uses the energy, the ATP energy from the cell, and produces ADP. So if we looked at the wild type, you produce ADP when you incubate it for longer times, but we don't when we have a mutant form. And this assay that we developed was amenable to high throughput screens to be able to find whether drugs could inhibit this activity. So what we've done since that time is perform a screen of about 120,000 compounds to look for molecules that can inhibit this function. And what we've been able to validate is that we can have 10 compounds, 10 known compounds that have already been have have other targets as well. They're not specific, but they at least do inhibit SMooCD1. And one new molecule that doesn't have anything else that's known about it, but we can tell that it inhibits SMooCD1 in this in vitro assay with the recombinant protein. So now what we're really keen to do is to do more medicinal chemistry on these drugs to try and get them to work better, more specifically, with higher affinity, and much more potently, so that we can take them into cells and see whether they have the same effect as when we knock out SMooCD1 or remove it entirely. So that's one way to hit SMooCD1, would be to have a small molecule that could be taken as a drug and really inhibit its enzyme activity. But what are there alternatives? So as a scientist, we always like to have plan A, B, C, D, maybe E and F up our sleeve, because A and B often fail, and so we want to still keep going through. So what are the other ways? And this is where I want to touch on just very briefly what it is that SMooCD1 does. So for a decade we've been trying to work out, what does it do? How does it actually switch genes off? And if we can understand how it switches the gene off, maybe that's how we can target it. So in this case, I need to tell you a little bit more about how the DNA is packaged in the nucleus. So we draw it as a single strand with a lot of genes along in a linear fashion, and that has got nothing to do with what it looks like in the nucleus. So here is actually, this is a nucleus, and each of the different colours is a different chromosome. So they have their own territories, they're compacted down. They first form these kind of, subcompartments. Then if you zoom in tighter, you see these topologically associated domains, and finally these loops. And the loops are really important for gene expression control, because you're bringing together regions of the genome that need to talk to each other. So all of our DNA is wrapped up like this all the time, and it's exactly how it's interacting which determines whether a gene gets switched on or switched off. So we thought maybe SMooCD1 was going to be involved in this for data that I won't go into. Maybe it might be involved in this higher order looping of chromosome structure. So in the same system I told you about before, where we can take out SMooCD1 for a week in culture, we did this genomic technique called in situ Hi-C, which analyzes chromosome conformation around the whole genome. And strikingly, what we found is for all of the places where we know SMooCD1 binds in these cells, we found a change in conformation everywhere. But remember, we didn't see a change in expression everywhere. We didn't see a change in expression for the Hox genes, the X chromosome. So this was kind of interesting. But the change in conformation everywhere told us that SMooCD1's function was being elicited in some way through mediating this higher order chromatin architecture. So we then relate this back to what we know at the Prader-Willi cluster. So there was a publication from more than 5 years ago now, where they found that on that inactive allele, this portion of the cluster that we work on that contains MAGEL2, NDN, and Macorin, that when it's off, it has a very long-range loop that's being mediated. It's over 1.4 million base pairs. And then on the active allele, it's doing something different. They didn't publish it, but we think it's actually being made into shorter-range loops that are helping these genes get activated. So from our data, what we find is that when we delete SMooCD1, we have a transition that's going from these very long-range loop into something that is very short-range, and which is consistent with sometimes activating those genes. Maybe not in every case, but activating that we can see and we can detect. So this means that another way that we could potentially target SMooCD1's role at maintaining imprinting at this cluster is actually not just to target the enzyme, but instead we could target the loops that it's forming. And this means that there are other genetic ways that you can do this rather than just targeting the enzyme. You can use technology that's similar to what others are using, where you can actually drive proteins to sit there and kind of get in the way of SMooCD1 where it likes to bind. So we're excited to think about these possibilities as well into the future. So at the moment, the next steps that we have for this project are we are generating the tools so that we can knock out SMooCD1 in human cells, so we can take patient-derived cells, and in particular, only knock them out once you get to the cell type of interest. So if we have pluripotent stem cells, we only want to knock them out once they become a hypothalamic neuron, because it's irrelevant if we knock them out of the equivalent that would have been a very early implantation embryo, because we're never going to do that in a human context. And in this way, we can check the expression of MAGEL2 in particular. We're going to do the same thing in an in vivo model where we can delete neonatally, again, because this is most relevant to the disease, we think. And then finally, the medicinal chemistry that I spoke about on those inhibitor molecules that we have. So I'd like to just acknowledge the members of my lab and, of course, all of our funding, particularly the Prader-Willi Research Foundation Australia, who've been able to fund this work as well. And Megan, who's here, who's done a lot of this work, along with Andrew Kennery and Natasha Jantz, who are fantastic people in the lab. Now, one past and the rest present. So thank you very much.
Thank you very much. That's really amazing work that's going on there. I can't wait to hear the next episode. And I'm sure there'll be a lot of questions for you and for the other speakers in our question time, which follows this session. So our final speaker for this afternoon, this particular session, is has been replaced actually. Kathleen couldn't make it, and we have got Emily, whose name is also on the abstract, Emily Meyer, and she's come also from the Foundation of Research Foundation. So thank you, Emily. Oh, that's right, there's two of you, isn't there?
I know it's the end of the day, but I feel very energised to have heard all of these incredible scientists and researchers and clinicians today. It's really, I feel very privileged to be in the room. So let me try and get this up first. That might be helpful. Working? Yeah. Sorry, I don't seem to have my mouse function. Oh, sorry. I just don't have the mouse. Okay, scroll on that. Oh, sorry. So yeah, my name's Emily. I'm speaking on behalf of Kathleen Jones today, who's the founder of our organisation, and she unfortunately can't be here today because her little girl Chloe is very sick. They've spent the last couple of months really exhausting and traumatising couple of months, as I'm sure most of you, all of the parents and clinicians would know what that's like. So she's unable to, she's trying to find out what's wrong with Chloe. They've been in the hospital. So I am the CEO at the foundation, and Diane Webster is our research director. We want to just tell you today a little bit about what we do at PWRFA, and we will talk about our program of work too. Firstly, thank you very much to the organising committee who have brought us here today, especially James. We owe a lot to PWSA in general, and also to the state bodies who have helped to fund the work that Marnie has done, that you've just heard a little bit about. So PWSA Victoria has helped us fund the first phase of that work, and PWSA New South Wales the second. So thank you very much for that. I also particularly want to thank the parents and family and friends. We are really deeply grateful to the parents that have been major funders of our work to date. We really greatly appreciate your efforts and take very seriously the responsibility to have the best outcomes for your children. To the parents in the room today, I'm really very conscious of even getting here is a big challenge, and it's a testament to your commitment to really understand and make a difference that you're here. And that's how PWRFA started, Kath wanting to make a difference. Kath asked us to share a little bit about her story as background for how PWRFA came to be. When Kath's beautiful little girl was diagnosed with PWS, she refused to stand by and accept her outlook. Kath's career was in organisational problem solving, so it made sense to apply her skills to Chloe and your children. After her diagnosis, Kath spoke to lots of scientists who told her that she shouldn't worry, that the biotools are available, give more hope than ever of a treatment. What PWS did and does need is a force to connect, to encourage, drive the agenda and attract funds to effect the change. Once you start talking with researchers, there are so many options to work on treatments, from improving growth hormone access through to constipation, exercise, appetite suppressants and genetic treatments that and that the real issues are how to attract enough funds, how to prioritise projects, and how to attract the most gifted researchers to PWS. So we were established to drive that research into PWS in Australia, and many of those researchers are here who are doing that, and it's really inspiring. We're certainly not alone in wanting to improve the lives of those living with PWS in Australia. We sit alongside PWSA and the Better Living, and of course the team who's put this who's helped to fund the program today as well, who I did not put this integration on there. So progress for a relatively small condition can only be made with collaboration and shared vision. And we're striving to strengthen our points of connection with these colleagues and to increase the pace of change. So this gives you a bit of an overview of our vision. The Prader-Willi Research Foundation Australia aims to transform the lives of people living with PWS and their families. We work to improve clinical outcomes and treatments for Prader-Willi syndrome so that those living with PWS can live full lives and fulfil their potential. And this vision is supported by a committed board of high-profile directors and a team of leading experts on our scientific advisory committee. And you heard from Professor Einfeld and Professor Herzog earlier today who sit with us on that. Diane and I work a limited number of hours, and we have a small team of dedicated volunteers, including Susan Blair, who's here today, who helps us very much. We're very aware that families have the most to gain from our success, of course, and it's only with family support that we can succeed in this. One of PWRFA's founding board directors is Professor Bob Williamson. He was running the Murdoch Children's Research Institute and is globally recognised for his work on cystic fibrosis. He taught Kath three important things. Firstly, that the outlook for people with cystic fibrosis has transformed due to the enormous amount of research, and this happened because of parents pushing and working hard to get money for research. Secondly, the transformation in life outcomes for people with cystic fibrosis was due to a number of incremental improvements. And you can see there, summing them up, that life expectancy went from 12 years to 40. And lastly, in the locations where research happens, clinical care is better. And this is because clinicians are engaged, curious, pushing the boundaries. These clinicians are focused 100% on one job, trying to solve the challenges of one condition. So this knowledge and experience of other similar conditions has shown that research can be transformative for current and future generations. So it's this advice that has helped to form the objectives of PWRFA. So this is what we aim to do: drive a program of research to materially improve life outcomes for people with PWS and their families, facilitate clinical trials of treatments that have a high potential to greatly improve the management of PWS, and Diane will talk to that in a little while, foster growth of the Australian Prader-Willi research community and their connection with clinicians and families, raise public awareness of PWS and funds for the research. So PWRFA's directed program of work has short and long-term outcomes. In the short term, we're looking to improve symptom management, developmental and behavioural outcomes, and longer term, the cutting-edge research to develop treatments to directly target the PWS genes, so specifically Marnie's work. So we'll achieve this by fostering deep medical expertise to improve management and funding research into breakthrough medical treatments that offer genuine promise and potential. So it's not just about the next generation and the long-term goals, it's really about this generation and short-term improving life as well now. We'll spend the rest of our brief time just telling you about the current program of work and our achievements. So this is where the funds raised are going. I'll talk about our foundational projects and healthcare healthcare delivery, as you see here on the screen, and then Diane will talk about the other parts. One of our key foundational pieces is to ensure that the Australian PWS community has a register. Registers are an important resource for understanding the depth and diversity of PWS. They enable the research and capture the voices of the PWS community. And our goal is to ensure that Australians with PWS have a high quality register that addresses locally relevant questions such as defining unmet needs and providing an evidence base for the NDIS. This is critical information to inform policy and practice at all levels, from defining our research objectives to clinical practice and government policy. We also need to ensure that we engage with international research efforts in terms of contributing to the overall numbers necessary to advance research for rare disease and to ensure that we have access to new information and opportunities. Over the recent months, we've worked with our scientific advisory committee, our independent registry experts and passionate and dedicated scientists at these institutions, and they have register projects that are either open or upcoming. The global PWS register started in 2015 and runs under the auspices of the FPWR in the USA. And a major focus for their register is the new PATH study, which has just been launched just a matter of weeks ago. That's a co-sponsored project by the Foundation and ZAFGen, the Foundation in the US, I should clarify. So the aim of that program is to better understand serious medical events in PWS over a 4-year period. And the data from this study is intended to inform the development and clinical trial design of potential new treatments. The Australian PWS register is being developed by the Telethon Kids Institute in Western Australia, with the goal of providing an evidence base for clinical practice in Australia. So they've put together a nationwide network of paediatric specialists and researchers at this stage, and they are in the process of doing that. They have ethical approval and it's not yet launched. But we'll certainly communicate about those projects as we know about them. So our other foundational pieces, once there is a register in place, we'll look at how we can link a biobank to that, because that again adds to the information that we have. There is a patient voices project that we're looking at doing potentially with PWSA. We are spending quite a lot of time trying to engage more with government. Okay. Oh, am I running really short of time? To try and push the investment from the government into PWS. Another one of our big projects is to try and engage a clinical fellow. So that's this idea of having a dedicated PWS clinician, and that's one of our key recommendations that we're talking to the Minister Hunt and Department of Health about. We really can't stress highly enough that having someone employed in a clinical setting who's focused 100% on Prader-Willi is key. And then in our healthcare delivery, we're also looking to do phase 2 of our healthcare model, which Kathleen has developed to try and drive that forward as well. So I'll let Diane talk to the rest of our program work.
It sounds like I'm doing the very short version.
Sorry. It's okay, I wasn't sure how long you were going to speak. So
Not
this
long.
is the second part of the speaker, the
Yes.
talk today.
So thank you, Emily. What I'm going to do is just give you the highlights of the second part of our program of work. So the first thing I'll talk about is access to new treatments. This has a very heavy focus on clinical trials. We are doing our everything we can to support local trials initiated by our world-class Australian researchers. We're also looking to attract some international trials. So this is the beginnings of what we hope will be a central point for clinical trials that are recruiting in Australia. I've spoken with a couple of researchers today who have trials not on this list that we'd like to see go up there, and we're keen to hear from any other Australian clinicians and researchers who are doing clinical trials so that we can do everything we can to help support and promote visibility of those trials. So some of these trials you've heard about today, the ENGAGE study from Amanda and Alex, from Stuart and Lauren, we've heard about the oxytocin trial. They have a mindfulness trial as well. There are also trials, for example, out of the Bearden Lab in the US, which are web-based. So this is a US study being done in the US, but anyone in the world can hop on the computer and complete their surveys, which are looking to eventually understand better some of the predictors of psychosis. There's a study in Queensland from that Miran Gu is undertaking for her PhD, which is looking at ways of measuring muscle tone, and the PATH study, which we've alluded to. I know from being here today that there's at least 2 or 3 others that can go on that list, and we'd love to talk more with you about that. In addition to supporting the local trials in any way we can, we are also talking with international companies, largely pharmaceutical companies, about Australia as a destination for, or a site for some of the larger phase 3 trials, where it's important to recruit large numbers to get the body of evidence necessary to answer the question of whether an agent's going to be safe and effective. So in the interest of time and not standing between you and your wine, the second 2 really kind of go together. They're our research focus. They're one of our major drivers to find, convince to swap over to attract our leading researchers in Australia into the field of Prader-Willi. Some are there already, and we're supporting them any way we can. Others we're hoping to attract to come and work on Prader-Willi. So we have a pipeline of projects. The latest one is from Nora Shields, who you'll hear from tomorrow. She's looking at resistance training. That project's been approved by a scientific advisory council and will most likely be something we're fundraising against in the near future. Of course, we're also incredibly proud of the work that Marnie Blewett's been doing on the MAGL 2 project, and the next step from our perspective there is to expand out that project so that we have more than one target looking at ways of addressing underlying genetic causes. And the final project I'll mention is a mapping project where we're looking at identifying the steps that would be necessary for epigenetic technologies to take them from the laboratory, for example, with Marnie's work, and actually what would be necessary to get them right through and actually take them to market. Because unless we can get things out of the lab and onto the market, then they're not going to have the positive impacts that we want them to have in day-to-day life. And so I hope that between Emily and myself, we have managed to give you a clear overview and an idea of the scope of the work that we're undertaking. It's critical that this research happens and that it finds its way out to the community so it can actually make an impact. What am I supposed to be saying? So yeah, what the Foundation has done is make a really solid start on this journey. We're looking to build our track record, which is strategically important so that we can attract the scale of funding that's going to be necessary to continue on this journey, a journey that we are sharing with you, and which keeps individuals with Prader-Willi and their families right at its core.
Please come and talk to us over the weekend. We'd love to talk to you more about what we're doing. Thank you.
Thank you very much, girls. That was lovely. Nice to know that there's going on. Now, we do have a question time for the panel. So do you want and you're going to chair that. So I hand over to the new chair and ask the panel of speakers today to make themselves available.
Yeah, thanks Ellie. Thanks, Ellie. Anybody who's still here who has presented today, could you just come up to the front? We are still going to have our question time, but we're running behind time, so we're just going to try and reduce them as much as possible. So perhaps if you just come to the front of the room, and I'll read some questions out and see who would be best to answer them. First of all, are there any questions from that last session that we've had?
Hi, I'm just wondering if the switching on and off of the genes explains the nutritional phases.
It's unclear. We don't know. It's a short answer. Everybody hear that? We don't know.
Okay, well, question number one is, can we get a copy of the slides, please? For anybody who doesn't know, this is being recorded on Facebook today, live, and there will be a recording up there which you can have a look at, which will include the slides. I don't know how clear they are, but if the speakers are happy to provide them, if you just provide them to the organising committee and we can forward them on to anybody who would like them. Question number two is, if you've got a crystal ball, approximately at what age does the hunger drive lessen in adults?
So it's variable. We've seen it in the 20s and 30s and 40s, but you know, it's not absolute, and it's not all the patients. It's only a subset, somewhere between 10 and 20%, and that's really the phase that we need to study a lot more. And I'm undergoing processes to do that, since I have 70 adults living in a residential facility in Gainesville. I'm working with the behavioral therapist there to really get at the right number and track that. And part of the problem with phase 4 is people say you live in a residential facility and everything's locked up. I mean, how do you really know about the hunger drive and the behavior modification? Well, there's several ways. There's individuals that still elope and go leave the home and go foraging for food. Also, most of the persons with Prader-Willi in the residential facility go home for periods of time, on vacations, weekends, whatever. And we track the weight when they leave the group home and when they come back. And I've had some individuals that, and we started doing that when I thought I had someone on the right diet prescription and they hadn't lost any weight. Well, it turned out the behavioral therapists were tracking it, and I got those graphs. And the patient would go home and over the weekend gain 10 pounds, and then over the course of the month would lose it 1 to 2 pounds per week. And when I added it all up, this person had gained 100 pounds and lost 100 pounds. So when they saw me back in clinic a year later, they were the same weight. So we can tell. Then there's some people that go home and the parents don't need to lock up foods. They don't need food security. And so some pretty interesting stories. So we don't really know, but you know, there's not one age that it happens. It's variable, 20s, 30s, 40s, but some people still in their 50s and 60s are still pretty food preoccupied.
Okay, this is a good question because this also applies to my son. This is for Stuart Einfeld. My son, who is 11 years old, PWSUPD, talks to himself and has a lot of imaginary friends, which is a lot like my son. It's all very cute, but at what point does it become a serious concern?
Okay, so that is not psychosis. The reason it's not, when it's psychosis, you notice a number of things. Firstly, you don't just get changes in the beliefs in isolation. There's a whole change in the person's coping function. So, you know, having beliefs which are, you know, not possible, or seeing things and hearing things which aren't there, are maybe the most striking symptoms, but they don't occur in isolation. So you get a whole cluster of disturbance, and the person is overall not functioning as well as they normally do. So, okay, he's 11, he's at school, he's participating in the class, you know, learning some new things and so on. That would be pretty young for the psychosis of Prader-Willi, but if it were, you know, then the child's whole behaviour and function, both at school and home and outside, would deteriorate. There may be sleep disturbances and so on. Then the nature of those phenomena is that you've described, imaginary friends, that's a fairly common phenomenon of childhood. And I forget what was the other?
Talking to themselves.
Well, talking to themselves. I mean, lots of people with developmental disabilities talk to themselves, and it's really just a socialisation.
I talk to myself all the time.
Yeah, that's right. We frequently talk to ourselves, but we don't do it. We usually don't do it aloud because we know that people will think we're peculiar. But people who don't have such social skills, don't have good social skills, may talk to themselves aloud because they're not so, alert to how other people might respond to that. So neither of those two behaviours is abnormal. Both in its nature and also the symptoms in isolation, not part of a whole syndrome of illness and deterioration which happens in the psychotic phenomenon. But it doesn't matter, you can keep an eye on it. If you're concerned, there's nothing wrong with, going to your doctor and asking advice about it, but that's what it looks like to me.
Sounds like good advice. Okay, now this is a really good one, and I'm sure lots of people want to know. In Australia, will adult growth hormone ever be suggested as a regular treatment for PWS who did not receive it as children, or even those who have received it as children? Who would like to take that one?
I'm not from Australia.
One from the foundation? Who?
There we are, Georgie, yeah.
I can just say something. We actually had a meeting yesterday. It was a brief meeting, and obviously the plan is to work towards that. There are studies happening in the Netherlands at the moment that are looking at growth hormone in adults, and we may be participating in one of those studies in Sydney. We've been included in their application, so we hope to start this study probably next year, and we're looking at growth hormone in growth hormone naive adults, so adults who have not had growth hormone before. But obviously the plan is to try and continue it for those who are on growth hormone at the moment. And in some cases it is being continued on compassionate grounds through Pfizer. So if your adults are nearing 18 years of age and they are on growth hormone, I would speak with your endocrinologists and work with them hard to try and put in applications for compassionate grounds to continue growth hormone. And this is happening at a slightly reduced cost. Is Deirdre here? Oh, she's gone. Okay. Okay. So remember, it's all about pushing and advocating, which we'll chat about a bit tomorrow. But yes, the hope is that it will eventually be available on PBS for children, for adults who have Prader-Willi syndrome, but not necessarily obviously meet the height criteria.
Okay, can we get a quick definition of the 3 subclasses of UPD? Now that's a good one because I didn't know until today that there was subclasses of UPD.
So there's 3 subclasses. One's heterodisomy. Can you hear me with this? Yeah. Doesn't seem to be on to me. But anyway, so heterodisomy, segmental isodisomy, and isodisomy. So mom has 2 15s that she can pass on, and if only one gets passed on and it gets duplicated, those 2 15s are identical. That's isodisomy. So we looked with Merlin Butler at over 100 UPDs, and about 10% are isodisomies. And we recently published in Journal of Medical Genetics 2018. The other type is segmental isodisomy, where the mom's 2 15s cross over during meiosis and they exchange materials, and then each of those 15s gets duplicated. But at any rate, if it's when in UPD where there's segments that are identical on these 2 15s, not the whole 15, it's usually 10 to 20 million base pairs out of over 100 million base pairs, but it's absence of heterozygosity, it's homozygous. And then the other type is heterodisomy, where you got the two different 15s from mom, there was no crossing over segments, and the 15s are totally different. The significance of that is that we're all carriers for 5 to 7 genetic diseases. And if you and usually it's silent. You don't know that you're a carrier for that unless you marry someone else that's by chance a carrier, like for cystic fibrosis or sickle cell or whatnot. And that's why years ago it was taboo in humans for close related marriages, brother, sister, first cousins, etc., because they noted the preponderance of increase in genetic diseases. So where this is significant in Prader-Willi is you could have a second disease because the mom was a carrier, let's say, for Bloom syndrome, and then those two segments are identical in the child with UPD. They could have Bloom syndrome by fact that it's been doubled now because there's two segments that are identical. So what we go SNP arrays, the chromosomal microarrays that have small nucleotide polymorphisms that should be different between the two different chromosomes. And it's called absence of heterozygosity or homozygosity. And so, again, the isodisomy is 10% in our study. The segmental isodisomy is 70%. And the heterodisomy, complete heterodisomy, is about 20%, 20 to 30%. Does that make sense, or
That sounds like a whole new presentation.
Yes, that's why I didn't talk about it too much. But if you really want to read about it, Journal of Medical Genetics, Merlin Butler is the first author, 2018.
That's great, thank you. Okay, how often should the IGF-1 levels be checked, and bone density, bone age, hand X-ray?
You need a pediatric endocrinologist over there. Come on, you.
Oh,
adopt you in.
From the consensus that they have, the most recent consensus, I think 2030, the IGF-1 was said to be checked every 6 to 12 months.
The one thing I've learned from the endocrinologists I associate with, IGF-1 tends to run high in Prader-Willi. So a number of endocrinologists like Matej Taubert, Maurice Angulo, Urs Holzer aren't as concerned as they used to be about having a high IGF-1. It's really tracking the mid-parental height, and I also track the head circumference. If someone's head circumference is greater than 95th percentile and everybody else in the family's parents and siblings are less than 50th percentile, that person's getting too much growth hormone, and you can get overgrowth. You don't think so?
No, I don't think they have high levels.
Huh?
I mean, I don't look after children.
No, this is after when they're treated with growth hormone.
Sorry.
So when you're treated with growth hormone, the IGF-1 for the same dose of growth hormone is going to be higher in an individual with Prader-Willi. Yeah, but before growth hormone, it's going to be lower.
I misunderstood.
Yeah. No worries.
You got a question?
No, I'm just trying to both of you that from the Australian database with PWS as a growth hormone, the doctors, it's a big collaboration nationally. They all try to keep the IGF-1 as much as possible within the appropriate range, but that's because we use a slightly lower dose than what they generally do in America and in Europe. Despite this, that we still get very appropriate growth and very appropriate metabolic BMI. It's now up to the endocrinologists themselves how much they want to dose in the new regulations that sort of came up since 2015, I believe. But yeah.
Okay, that's great.
That was the situation.
Thanks, Ellen. Okay, is premature adriarchy the same as early onset of pubiarchy?
Premature adrenarchy is basically your adrenal glands get stimulated earlier. Premature pubarchy just means that the pubic hair comes out earlier. Pubarchy is part of adrenarchy. So when we say adrenarchy, that means there is acne, there probably the body odor together. It comes earlier. Usually when we talk about puberty, in girls the breast should start first, and for boys the testis size increase. If let's say you have the body odor that comes earlier than the testis enlargement, so you call that as premature adrenarche. If the pubic hair starts earlier than the breast development, that's premature pubarche. So that's a different. Premature pubarche is when pubic hair comes earlier than the breast, because in girls the breast should start first. Premature pubarche is not a true puberty. Premature adrenarche is also not a true puberty. True puberty for girls is breast development first. True puberty for boys is when testes increase in size. But usually, even for kids and for anyone, if you don't check the testis size, you don't know that the boy has gone into puberty. Yeah.
I think that a lot of children have adrenarche first. I thought that often
the natural history that the adrenarche and pubarche should come after. In a true puberty, they have to follow the natural history. The natural history is that for a girl, breast should come first, then only your pubic hair and the other adrenarche, the body odor and all that. For boys, testis size increase first, then only you have the pubic hair and also the body odor. If those things are reversed, that is not true puberty. Yeah. From the thing that we know under the pediatric endocrinology in terms of puberty.
Okay, now this one is interesting. As a layperson, I didn't think we advocated, but for males born with PWS, is there any research on information around the benefits of circumcision?
Well, it depends on how big the penis is, right? The phallus. So that's the whole circumcision thing's rather controversial, and that's a personal choice. Some people think it reduces UTI, but there has to be a certain size to the phallus to do circumcision. So I think if people feel comfortable doing it, and that's what they the family wants, or their religious beliefs, then go ahead and do it.
And this was for Margaret Harris, but I don't know whether anybody else might be able to answer it. If a child with PWS has two older siblings with narcolepsy, can I expect an increased risk of that child to developing the same thing in adolescence?
Unless someone else wants to answer. As a geneticist, I would think that would be increased because there are certain genetic factors. So yeah, I think it would be increased if talking about a true case.
Are there any questions from the floor?
A few. Now the first question is for Dan Driscoll. You mentioned really shortly about skin picking and N-acetyl. And I've just personally never heard of the combination.
N-acetylcysteine?
Yeah.
Oh yeah, we use it a lot. Pharmanac is one of the drug names, and there's other. You can get it in a pill form, but the form I prefer is like a tablet you drop in the water and it fizzes, and you drink 8 ounces of this like an Alka-Seltzer. And actually, Jen Miller and Morris Angulo published an article about it. So I initially had collaborated with Andy Shapiro on the topiramate and low dose, and I think Stuart maybe mentioned that earlier. A much lower dose of topiramate than you'd use for seizures, 25 to 50 milligrams. And Andy originally was doing it to see if they'd change an appetite, and what he found, it didn't really change the appetite, but it reduced the skin picking. But this, topiramate is a pretty strong drug. And then I think Jen Miller was informed by some of the parents that, hey, Pharmanac works pretty well. So she did a trial with Morris Angulo, and that seems very effective. I think it's about 70 to 80% of the patients I notice a difference in, and I use it quite a bit. and I think that the pyramid is maybe 40 or 50%. And sometimes one drug will work and the other one won't, and sometimes you use a combination, and there's behavior modification. But the Pharmanac, I think, is 70 to 80% efficacious. Sometimes it eliminates it, sometimes it reduces it. But you can get it online. It's over the counter, so it's not a prescription. And there's a website. You just Google Pharmanac.
And from what age?
From what age? Well, skin picking, once I start seeing skin picking. And it's a pretty safe drug. I forget the exact mechanism, but at one time I knew it, but I've since become senile and lost it. But it seemed pretty safe to me. Not real. And it originally was used for trichotillomania, where people sit there and pull their hair out. When I was a medical student, my wife and I, there was a girl in front of us that she'd do it in all the lectures. And I wish I could go back and prescribe this drug.
What doses are you using?
The Pharmanac is 900 milligram tablets, and I start once a day. And if I don't see an effect or I don't see the total effect, then I do it twice a day, once in the morning and once at night. And you do have to drop it in the water, 8 ounces, and then drink it.
And what happens if you get consistent results that it's easing the skin picking, but then all of a sudden it picks up again? Because I've found having NAC quite inconsistent. Steph goes for a period of absolutely no skin picking, all of a sudden it flares up again.
So Jen Forrester and Linda Gurash, who some of you may know, do a lot of behavioral stuff, would say, I'm channeling their voice now, that check to see what else is going on. There may be some stressor in the family, like the mother's pregnant and there's going to be a new baby in the family, or there's parental. Are you sure? Have you had a pregnancy test? Or some other stress if the parents are breaking up. So first check to see if there's something in the environment that's changing. And then, once I check everything out, I may add topiramate on top of that. Of course, I check with the psychiatrist to make sure, it's not conflicting with something else they're doing on some of the adults. On the young kids, they haven't gotten to the psychiatric meds yet.
So if you do the Pharmanac, is this something you do all the time, or only when the skin picking starts and then for a while and you stop it, or?
Well, I see how bad the skin picking is. I talk to the families. Roughly about 50% will skin pick and 50% won't. And I haven't totally drilled down on the numbers, and we haven't quite figured out why some do and some don't. But, that's the way genetic diseases are. There's variability. But, the skin picking has to be significant, not just a little. And I've actually used it in some of my other genetic conditions where there's some skin picking, and suggested they try it. It's used a little bit anecdotally, so I don't have a lot of data on that. But on the Prader-Willi syndrome, I think it's 70 to 80%. In Jen and Morris's article, it was like 100%, but nothing's really 100% when you increase the numbers.
Question? Yes? Good though.
Just in regards to the research that you're doing, would most cases of UPD then start as mosaicism? And is your testing specific enough to pick up all of those cases?
Can you ask an easier question? No, so by mechanism of UPD, there's first trisomy with meiosis, and then for it to be viable, there's this thing called trisomy rescue, so the other chromosome is thrown out. So that's how UPD originates. That's the main mechanism. But in a proportion of cases, at a low level, some cells do not throw out that chromosome. We've got preliminary data to suggest it might be around 10%, but don't quote me on this as yet because the study is not finished. And how low can you go? We're all mosaic for trisomy, or at a very low level, we're all mosaic for some mutation. So the question that we're asking now, how low is clinically significant? because we're not looking in the brain, we're looking at peripheral tissues. So to some level, it might be conserved between tissues, but if you go too low, it probably doesn't do anything. That's why we want to compare the ones that have features, phenotype, intellectual disability, or other features that have been recognized by clinicians to have issues that overlap with Prader-Willi versus general population, where majority don't have this. So if you show the difference that is statistically significant, you can say that at that particular level it is clinically significant. So if you have very sensitive test, doesn't necessarily means it's good. You just need to know how low you want to go. And we don't know yet.
Okay, just a quick follow-up. You said the numbers are, you presume they're vastly underrepresented. I know you probably don't want to throw a number out there, but do you have an idea of a number?
That's a very interesting question. So, everyone's, from US especially, have been talking to the first thing they say, so what's the prevalence in your studies? And my answer to them is, I'll give you a prevalence before it's finished, and then I'll be wrong when it's finished, because we haven't done the full cohort. The plan is we do probably 20,000 published and do another 20,000 published to see whether it's reproducible. I think it's below 15%, 1 in 15,000 for sure. And I think our challenge is newborn blood spots. G15Q is a lot more common, and it comes up at about the same level of methylation, maternal G15Q, as those UPD trisomy 15s, as imprinting disorders. Another issue that I don't think has been, I'm new to the Prader-Willi field, but I think one issue that hasn't been discussed from what I've read in too much detail is that if there's an epigenetic event happening elsewhere in the genome, and that's stuffing up methylation everywhere, but you're only looking at the chromosome 15, so it might be stuffed up in other loci that are epigenetically dysregulated.
So 1 in 5,000.
So I think I can't say what it is, but it could be twice as common or 3 times as common. But those cases may not be classical Prader-Willi, as some of the ones that I said we have actually identified through dev delay referrals. They were diagnosed at 20 years of age 2 years ago. We have one that was diagnosed at 20 years of age, intellectual disability, severe intellectual disability, not like Prader-Willi.
We have a few of those in Asia.
Short stature, never had problems with appetite, and various other unusual things. So it came through microarray testing or through the dev delay exome sequencing referrals. And we had a case, one case collected that prenatally showed trisomy 15, and after birth they couldn't see it anymore. Does not mean it's not there. It might not be there in the tissue test, it might be elsewhere. The trick is to see how does it reflect functional parameters in terms of gene expression in different tissues and the phenotype as well. So that's mine.
Okay, in the interest of time, we've probably got time for 1 or 2 more questions. So we've got one up the back.
Hi. I just wanted to ask about rectal picking. Is it common? What can be done? And how serious can it get?
Well, I think of rectal picking as the same phenomenon as skin picking, and it's just a variety of skin picking. So I would tend to treat it in the same way. Sometimes, there are, strategies like if you have to go to, overalls or something, so the person, it's a physical barrier to rectal picking. It can be, it's obviously a upsetting kind of a behaviour and bothers everybody around. I don't know in terms of sort of surgical injury that to people, I don't know whether that happens. I don't know if Dan's had that experience.
Yeah, we've had some that have done so much that they've had a rectal prolapse from the rectal picking. It's not as frequent as the regular skin picking, or at least, could be underdiagnosed. But I agree with Stuart's assessment. And we would think that the Farmanac or N-acetylcysteine would help that. I haven't done a long study of that because, you know, out of, let's say, the 100 adults I follow, not, at least that admitted it, it's, you know, maybe 5 or 10 do rectal picking that it's admitted to, whereas 50% do skin picking. But I think it's the same mechanism as skin picking.
Okay, next one, Dolly.
So in terms of diagnosing the Prader-Willi, I recently met a mum who had a child age 6, presented with all of the symptoms of Prader-Willi, but testing had been said to be negative, so she was struggling to access services, etc. So yes, we've heard today about the Schaff-Yang syndrome, but what to do when we do come in contact with a cohort where the tests are negative?
Exome sequencing. Send it to exome sequencing. Exome sequencing.
Sorry, I can't hear.
Exome
He's
sequencing.
saying whole exome sequencing, so sequencing the whole genome. There's other conditions that are Prader-Willi-like, and you know, it depends on who's really seeing the individual. Schaff-Yang has some features like Prader-Willi, but they have arthrogryposis, you know, stiff joints that can't move and much more artistic. You don't get the obesity that Prader-Willi individuals do. There's a SIM1 deletion that can be Prader-Willi-like. One of the speakers talks about uniparental disomy 14. So there's various phenocopies you have to look. Bardet-Biedl sometimes is I had one patient that was diagnosed as Prader-Willi that had Bardet-Biedl. The thing is, the punchline is you have to look for another genetic etiology. If they've seen a clinician that's very familiar with Prader-Willi. Now, I've had sometimes the testing has been wrong. DNA methylation analysis, David talked a bit about the methylation-specific PCR, and I talked a bit about it. There can be errors in there. If there's a polymorphism in the primer that you use to amplify up the SNRPN CPG island, you may get a false positive. I've had a patient that I can't explain that had University of Connecticut had normal methylation, and at LabCorp had normal methylation, but a microarray showed a big deletion. So they came down and saw me, and I did MSMLPA on there, which tests 5 different sites, and it was Prader-Willi. So sometimes there can be a laboratory error, and the methylation-specific PCR, it's very sensitive to contamination. If the tech gets some of their cells mixed in with the patient sample, it's going to amplify it up. And speaking of the mosaic trisomy 15 in UPD, I presented that earlier in my talk, and David talked a bit about that. That patient had normal methylation test. Because there was 16% of the cells, it turns out, with trisomy 15. And so there was, you know, a paternal and a maternal contribution, and methylation-specific PCR amplified it up. And it's not very good at quantification. It's usually a yes or no answer. Is there a maternal allele? Is there a paternal allele? But if there's a low-level mosaicism, you know, with a normal cell line, or with, you know, both a paternal and maternal, it'll amplify it up. So there's several different possibilities. And you know, the other thing is the methylation will not pick up the SNORD116 deletions. So you want to have a chromosomal microarray. And the SNORD116, they're very Prader-Willi-like, but they're not, in my mind, classical Prader-Willi. Because those other genes are doing something, nectin, macaron-RN3, MAGEL2. So they need to, first of all, see a geneticist that's very familiar with Prader-Willi. And then probably do additional testing if that person's comfortable that it's close but not quite.
Okay, last question, I think.
Just one in relation to epigenetics, and I won't use the correct terminology, but if you were to switch on a gene, at what stage clinically could you use that with someone with Prader-Willi? Like, is it a neonatal thing? Is it a thing as a child? Fast forward into the future years when you're actually able to switch on the methylated genes.
Well, I guess the earlier the better, because many of these things are causing developmental processes that probably upsetting them. Well, we don't know exactly, but that's what normally happens in that thing, which would normally cause a different development of the hypothalamus. In the very rare situations where we have, for example, a leptin deficiency, that's a very rare thing, but where the people have no fat signal back to the brain and tells them, these people, you know, they're 80, 90 kilos at 5, 6 years of age. If you give leptin back, their hypothalamus changes again and they're completely normalized. So there's a lot of plasticity in the brain that can adapt to it. So the earlier you can get in, obviously the better. So I mean, there's a caveat to all this turning on. We have about 100 imprinting centers in the genome. So there's about saying, so really to find one that just turns on the missing Prader-Willi one will be still quite a challenge. And I think there has to be still a lot of things to be done to make sure that you really get the right one as well. But it's technically possible, and there's much more things going on. There's another technology kind of called CRISPR-Cas9, which you can actually manipulate the DNA itself. So there will be lots more research going on, and as I think what I can see from the literature I am working, there's an expanding interest in that, and I think there will be much more happening in the future.
Thank you.
Okay, I would just like to say thank you. Oh, sorry.
Just a quick comment about this. I think from natural history studies, with mosaicism, there might be something relevant to reactivation. Because in those, in that one case, I mean, it's preliminary, that one case that had close to 10% mosaicism of the third chromosome that was active, did not have any appetite problems, as I said. So you might not need to have too much of active to correct some of the things. You might not need to go to 50%.
But just a quick follow-up. What is, as a layman, what does it physically look like? Is it a surgery? Is it an injection? What would it look like in the future to actually revive a?
Well, as I said, there are different approaches you can do. If you target one of these methyl transferases, which modifies these histone molecules, you can come up with a drug. You can just give a drug in a sense. The other mechanisms, if what we're using is like what's called more gene therapy approach, where you take a virus and inject it into the cells and they produce the missing gene. So there are varieties of things that can be done. So it depends a little bit on what you kind of want to do.
Okay, I would just like to, as a parent of a son with Prader-Willi syndrome, it gives me great hope for all of the research that I've learned today. These days of the science, while it's a lot of information to take, it's very comforting to know that there's a lot of research out there, and there's a lot of hope. So I would like to thank all our speakers for coming along. Okay, so dinner tonight is going to be held in the Chelsea room from 7 p.m. Those of you who have got dietary special considerations, if you haven't seen the kitchen, please make sure you do so.